There's a heist that happens on the ocean floor, and it's one of the most complete takeovers in the natural world. It begins with a crab just going about its day. Then something tiny, almost invisible, settles onto its shell. It's a larva. But it's not looking for a home, it's looking for a victim. It finds a weak point in the crab's armor, a joint in its leg, and then it does something unbelievable. It injects its entire soft body directly into the crab's bloodstream. From that moment on, the crab is no longer its own. Inside, the parasite grows a network of root-like tendrils that wrap around every organ, infiltrating the nervous system. It chemically castrates its host, stops it from growing, and begins to completely rewire its brain. The crab is now a living puppet, a bodyguard and a nanny, forced by an unseen will to protect and nurture the parasite's children as if they were its own. Its body is just a shell, driven by a different mind. And the mastermind behind this complete biological hijacking, the creature that has turned this crab into a zombie, it's a barnacle. Welcome to the deep dive.
Yeah.
Wow. That opening, I think, really sets the stage for the incredibly strange and just endlessly fascinating journey we're about to take today. We're plunging headfirst into a world where biology gets twisted into forms that are so unexpected, so bizarre, they genuinely challenge our deepest intuitions about what it even means to be alive, or maybe in some cases, truly undead.
Right. Not quite alive, not quite dead, something else entirely.
Exactly. We're talking about the unexpected, the mind-bending, and frankly, some of the most outrageous adaptations nature has ever cooked up. Our mission, as always, is to kind of navigate this weirdness for you, digging into the research and pulling out the most astonishing facts and insights.
The wild ride.
It really is. So let's unpack this right from the start. Our human intuition, the gut feeling that helps us with everyday stuff like deciding if food's gone bad or when to cross the road, it often falls spectacularly short when we try to apply it to these huge, complex, natural processes.
Oh, absolutely.
And nowhere is that clearer, I think, than when we're wrestling with things like natural selection and just the sheer relentless grind of evolution over geological timescales. We tend to bring our own mental shortcuts or human ways of thinking to the table, and sometimes, those shortcuts lead us way off track.
That's spot on. Our brains are, beautifully adapted for immediate things, tangible outcomes. We look for clear intent, a straightforward cause and effect that happens, within our lifetime, preferably faster.
Right. We want the simple story.
We do. So when you encounter processes like evolution, unfolding over millions, even billions of years, or processes that are non-teleological, meaning they don't have an inherent goal or purpose, no conscious direction, our intuition it struggles to keep up. It's like trying to grasp the power of compound interest just by looking at one day's interest payment.
Yeah, you see a few cents and think, so what?
Exactly. You see that tiny fraction and your gut tells you it's nothing significant. But over time, as you know, it becomes absolutely astronomical. Think about that classic story, maybe it's true, maybe not, about the chess master and the emperor.
Oh, the grains of rice on the chessboard?
That's the one. The master asks for this seemingly tiny reward. 1 grain of rice on the first square, 2 on the second, 4 on the third, doubling each time. The emperor, thinks, sure, easy, probably feels generous.
He thinks he's getting off lightly.
He does. But by the time you get halfway across the board, let alone to the 64th square, the amount of rice needed is, it's more rice than exists in the whole world, probably.
Yeah.
Even a powerful emperor gets totally fooled by simple exponential growth because our gut instincts, psychologists call them heuristics, basically mental shortcuts. They can completely mislead us when we face these compounding forces.
That is such a perfect analogy. It really shows how those shortcuts just derail our understanding when things happen incrementally over vast periods. And that, I think, ties directly into one of the biggest misunderstandings about natural selection. You often hear it talked about it's some kind of intelligent designer, right?
Yeah.
It's got this quality control department meticulously crafting perfect creatures.
That's a really persistent idea.
Yeah.
But let's be super clear here. The ruthless inspector or the quality control department in evolution isn't some conscious designing force. It's simply death.
Oof.
It is brutal. Death and the constant, unending possibility of not reproducing. Natural selection just eliminates anything that doesn't work well enough to survive and reproduce compared to whatever else is out there in that specific environment at that specific time.
So it's not about being perfect, just better than the other guy?
Pretty much. Or just good enough to hang on. This process, over long enough time, makes things look incredibly designed, almost perfect sometimes. But it's actually just the result of relentless, undirected sifting. Generation after generation. It's not striving for some ideal. It's just about what manages to stick around and pass on its genes right now. No plan, just endless trial and error, and the errors get wiped out.
And speaking of what manages to stick around, that brings us straight to this really fascinating cognitive bias called survivorship bias. This is where we tend to only focus on the things or people or organisms that made it through some selection process, and we completely ignore the ones that didn't make it.
Right, because the ones that didn't make it aren't around to be studied.
Exactly. The classic example, probably the most famous one, comes from World War II. Statisticians were trying to figure out the best place to add armor to bombers that were returning from missions over enemy territory.
Okay.
Now, the initial, very human, very intuitive reaction was, look where the returning planes have the most bullet holes.
Right.
Wings, fuselage, tail, those are the parts getting hit the most, so armor them up. Seems totally logical.
Yeah, that makes perfect sense on the surface. Protect the damaged bits.
But this is where the cold, hard logic of statistics just sliced right through that intuition. The analysis, led by Abraham Wald, famously recommended the exact opposite. He said, no, you need to armor the places that don't have bullet holes.
Wait, the places that weren't shot. Why?
Because the very fact that those planes returned, even riddled with holes in their wings or tail, meant that damage to those areas wasn't catastrophic. It didn't bring the plane down.
Ah, I see. If a plane got hit badly in, say, the engine or the cockpit,
it wouldn't come back. You'd never see those bullet holes because the plane and its crew were lost. So the bullet holes you do see are on the parts of the plane that can take damage. The real vulnerabilities are the spots on the returning planes that are clean.
Wow. That's brilliant, actually.
Yeah.
Our intuition, focusing only on the survivors, gets it completely backward.
Totally backward. It's such a powerful reminder that what we perceive as working or what we even perceive as existing is often heavily skewed by what managed to survive the filtering process.
And that is a fantastic lens for looking at evolution.
Right. It's basically billions of years of survivorship bias written across the entire tree of life. It's just incredibly hard for us to grasp because, like you said, we're not wired to think about what countless tiny incremental steps playing out over millennia, over millions of years, can actually achieve. It's not about reaching some ultimate perfection.
Not at all.
It's just about what was good enough in that moment, in that environment, to keep the genetic ball rolling, so to speak.
Precisely. Evolution doesn't create optimal designs like an engineer would. It's deeply scrappy. It's opportunistic. Whatever is good enough to outcompete the alternatives right now. Whatever lets an organism survive and reproduce just a little bit better than its neighbors. Yeah. That's what gets passed on.
So organisms aren't perfect machines then?
Far from it. They function, often quite adequately, sure, but they're carrying what you could call a giant train of historical baggage.
I like that.
Yeah. Baggage and imperfect compromises build right into their bodies and their DNA. Think of them like old houses that have been renovated and added on to over centuries. They're full of quirky angles, weird plumbing runs, maybe some drafty windows, but somehow they keep standing. That historical baggage can even limit future options sometimes, but it also shows the amazing resilience of just being good enough.
And sometimes that baggage or just our interpretation of what we see leads to really fascinating misunderstandings, like the whole idea of living fossils. Remember the coelacanth?
Oh yeah, the famous fish from the deep.
Exactly. When the first living one was caught off South Africa in 1938, marine biologists were absolutely stunned because this thing looked, at least superficially, almost identical to fossils from the Cretaceous period, the age of dinosaurs.
Right. It looked like it hadn't changed in, hundreds of millions of years.
That was the assumption. Everyone thought it was basically a creature frozen in evolutionary time, a relic, unchanged.
And there's that intuition again, isn't it? Judging a book by its cover or a fish by its fins. Because genomic studies, looking at its DNA, have completely shattered that idea. Coelacanths have never stopped evolving.
Never. So they've changed.
Oh, massively. While their external shape, their morphology might be remarkably conservative, it hasn't changed much visually their insides. Their organ systems, bone structures, their cellular processes, their genes, they all show huge amounts of evolutionary refinement and change over those hundreds of millions of years.
So the outside looks old, but the inside is constantly being updated.
Pretty much. That conservative body shape isn't evolutionary stagnation. It's actually an incredibly successful solution for their specific deep-sea lifestyle. It works, so why change the basic blueprint? But everything under the hood, so to speak, gets constant tweaking and upgrading. There was one paper that put it quite bluntly, saying something like, the coelacanth morphologic conservatism is a belief not based on data.
Wow. So it's just a superficial observation that became dogma?
Essentially, yes.
Yeah.
And you see similar things, by the way, in groups like sharks and crocodiles. They often have these stable external forms that look ancient, but genetically and internally, they're constantly evolving and adapting. They just found a winning body plan early on and kept refining the details.
It's such a good reminder that what you see isn't always what you get in nature, which brings us neatly to another area where appearances can be incredibly deceptive: mimicry.
Ah, yes. Nature's copycat.
Right. Most of us probably learned about the monarch and viceroy butterflies in school.
Yeah.
That classic textbook example of Batesian mimicry.
Where the harmless one copies the dangerous one.
Exactly. The story went, the harmless viceroy copied the toxic monarch butterfly to trick birds into leaving it alone. Seems simple, elegant, made perfect sense.
A classic case study, taught for decades.
Until 1991. When David Ritland and Lincoln Brower published this paper in Nature that just, well, it blew the whole thing up.
Yeah, that was a big one.
They revealed that, actually, viceroys are just as unpalatable as monarchs. Both species are pretty nasty tasting to predators.
Right. Turns out they get their toxins from different food sources as caterpillars. Monarchs from milkweed, viceroys from willow leaves, which contain salicylic acid, basically, aspirin.
So they're both toxic, just in different ways. Which means it's not Batesian mimicry at all.
Correct. It completely reclassified their relationship as Mullerian mimicry.
Yeah.
That's where two or more unpalatable or dangerous species evolve to look similar.
Why would they do that?
It's like sharing the advertising cost. If both toxic species have the same warning signal, say, bright orange and black wings, a predator only needs one bad experience with either species to learn to avoid both. It benefits everyone involved, reduces the number of individuals that get sacrificed during the predator's learning curve, like two different biker gangs wearing the same "don't mess with me" colors.
That makes sense. Share the burden of educating the predators.
Precisely. And what's even cooler is how flexible these systems can be. More recent research by Kathleen Prudhoe, for instance, has shown even more nuance. Like, in areas where queen butterflies, which are another toxic model that viceroys mimic, become rare, the local viceroys actually become more toxic themselves.
They adjust their toxicity levels. How?
It seems they can regulate it based on the local conditions, predator pressure, the abundance of their co-models. It just shows how evolution can create these incredibly flexible partnerships and adaptable strategies without any conscious planning, no committees involved. It's just this continuous dynamic feedback loop where whatever works best right here, right now, gets reinforced.
It's amazing. Okay, so we've talked about how our intuition can kind of trip us up when thinking about evolution's clever, sometimes counterintuitive tricks. Let's bring it back now to the creature that kicked off our whole discussion, the one behind that underwater heist, the barnacle.
Ah, yes, the barnacle. Not usually seen as a master criminal.
Right. Most people think of them as, well, just those hard, crusty things stuck on rocks or maybe boat hulls or sometimes even whales. There's a great quote from Louis Agassiz, that pioneering scientist from the 1800s. He described barnacles as nothing more than a little shrimp-like animal standing on its head in a limestone house and kicking food into its mouth.
That's still a pretty accurate and evocative description, actually. It's remarkable he figured that out back then, given how unshrimp-like they look on the outside.
Totally. I mean, you look at a barnacle, your first thought is probably mussel or clam, right? Some kind of mollusk.
Yeah, intuitively they seem more like shells, but Agassiz nailed it. They are crustaceans, distant relatives of crabs and lobsters. And they really do feed by sticking their modified legs, these feathery things called cirri, out into the water and kicking food particles towards their mouths. So yeah, the image of them standing on their heads, kicking food in, it's bizarre, but it works if you're glued down.
Like little lazy perverts, as you said. Glued down, waiting for dinner to drift by.
A highly effective strategy for a sessile life.
And speaking of bizarre, if you've spent any time looking up weird animal facts online, you might know about the barnacle's other claim to fame, their reproductive anatomy.
Ah, yes. The penis-to-body ratio.
It's legendary, largest in the animal kingdom, right? Up to 8 times their body length. That's
Right.
quite something.
It is quite something. But while it sounds like, some kind of evolutionary showing off, it's purely practical. Think about it. You're stuck to a rock. You can't move to find a mate.
So you need reach.
You need exceptional reach. That incredible appendage allows a barnacle to reach out across the water and find neighbors to inseminate. It's a solution to a fundamental problem of sessile life. And it gets even more interesting.
How so?
Research by Christopher Neufeld and Richard Palmer show that barnacles can actually adjust the size and shape of this organ depending on the water conditions.
Seriously, they can change its shape.
Yep. In rough, high-wave environments, they tend to grow shorter, stouter, more robust organs, less likely to get damaged by the waves. But in calm, sheltered waters, they grow longer, thinner ones to maximize their reach.
Wow.
They even documented barnacles changing their anatomy when they were transplanted between these different environments. It's a fantastic example of environmental plasticity, the organism physically altering its form in response to its surroundings to boost its chances of reproduction, all unconsciously driven by selection.
That's incredible adaptability. Now, most barnacles, despite their unique anatomy, are pretty harmless to the things they attach to, right? Like on a whale or a turtle, it's usually commensalism.
Generally, yes. The barnacle gets a ride and a feeding spot. The host is mostly unaffected. It's a common, usually benign relationship.
There's always a but in biology, isn't there?
There often is, and in this case, it's a big one. There's one particular group of barnacles that completely throws that benign relationship out the window. They're not just hitchhikers. They're the hitchhikers from hell you mentioned earlier.
This is where we get back to our heist.
This is where we get back to the heist. This species doesn't just attach to the host. It invades and consumes its very identity.
Okay, take us there. Paint the picture. In the hushed blue light of the marine biology lab, long after everyone else has gone home, I watched Tank 7B. Inside, a green shore crab, a male, meticulously tends to a bright orange sac beneath its abdomen. It cleans the sac with a delicacy I've only ever seen in egg-bearing females, protecting it from debris, aerating it with its legs, almost fanning it with care. But the sac is not its own brood. It is the external body of Seculina carshini, a parasitic barnacle that has devoured the crab from the inside, castrated it, and hijacked its very being. The crab's mind, its instincts, its parental drive all have been rewritten to serve the parasite. I feel a profound unease, a cold dread that has little to do with detached scientific observation. I see a loss of self so absolute it borders on the metaphysical. The crab is a ghost in its own shell, its body a vessel piloted by a stranger. And in the quiet hum of the water filter, I can't help but ask myself, what is the I that I cherish so dearly if it can be so completely and utterly erased? That's the chilling reality of Seculina carshini. Its name literally means little bag of the crab. And it starts its life looking like a normal free-swimming barnacle larva, tiny, looking for a host.
But not looking for a rock. It's looking for a crab.
Exactly. And when this female larva, let's call her GLaDOS again for her ruthless efficiency, finds an unsuspecting crab, she does something truly nightmarish. She molts into this specialized, incredibly invasive form called a kentrogon.
Which is basically just a living hypodermic needle.
Pretty much.
Right.
A hypodermic needle made of flesh. And it finds a weak spot in the crab's armor, maybe a joint in a leg, or near the gills, and then it injects its entire soft body directly into the crab's bloodstream. Just poof, gone inside.
That single injection, that's the start of it all. The moment that initiates what scientists have called the most complete hostile takeover ever documented in nature.
A complete hostile takeover, and the result is, well, it's hard to even describe.
It really is. One paper put it chillingly perfectly. After infection, the crab has the phenotype of a crab, but the genotype of a barnacle.
Meaning it looks like a crab on the outside.
It looks like a crab. It has the physical body of a crab, but its internal programming, its biological operating system, is now being run by the barnacle's genes. The barnacle achieves this by growing this incredibly intricate, almost invisible network of root-like tendrils inside the crab.
The interna.
Exactly, the interna. These tendrils spread throughout the crab's entire body cavity. They wrap around every organ, they infiltrate the nervous system, and they physically and chemically rewire the crab's biology to serve only one purpose: the barnacle's survival and reproduction.
It's like microscopic ivy taking over a house from the inside.
That's a good analogy. And the physiological changes are drastic. The crab stops molting so it can't grow any bigger. It becomes permanently infertile. It can't reproduce for itself anymore.
Castrated by the parasite.
Chemically castrated, yes. And perhaps weirdest of all, if the host crab is male, it gets feminized. It actually develops female characteristics like a broader, flatter abdomen.
Why does it do that?
Because that broader abdomen creates the perfect sheltered pouch underneath the crab, exactly where a female crab would normally brood her own eggs. But instead, it becomes the nursery for the parasite's external reproductive sac, the externa crab, which eventually emerges.
So the male crab is turned into a pseudo-female to carry the barnacle's babies.
Precisely. But the truly mind-blowing part, the part that feels most like science fiction, is the behavioral hijacking.
This is the nanny and bodyguard part.
This is it. When the parasite's eggs inside that external sac are ready, the crab, male or female, it doesn't matter anymore, performs these elaborate maternal behaviors. It will climb to high points on rocks, just like a female crab looking for a good spot to release her larvae into the currents.
It performs release dances.
Yes, these rhythmic fanning motions with its legs, presumably to help disperse the Sacculina larvae, and it diligently cleans and protects that orange sac, the externa, grooming it, defending it, treating it exactly as if it were its own precious egg clutch.
It's become a living puppet, its own instincts completely overridden.
Completely.
Yeah.
A puppet, a bodyguard, a nanny. Its entire existence is now dedicated to the barnacle's reproductive success. It's just an empty shell driven by the parasite's will. That question you posed earlier about the nature of the self, it hits really hard here.
So how? How does it achieve this level of control? Is it just chemicals or?
It seems to be more direct than just chemicals floating around. Advanced imaging, like micro-CT scans, has shown those root-like tendrils, the interna, are concentrated really heavily around the crab's hepatopancreas.
Which is like its liver and pancreas combined.
Right, so that concentration is probably for sucking up as many nutrients as possible. The parasite is literally feeding off the crab's core energy reserves. But crucially, the scans also show these tendrils extending directly into the crab's brain and its central nervous system.
Direct neural infiltration.
That seems to be the key. This direct physical connection to the nervous system is the most likely explanation for how Sacculina can pull off such dramatic, complex manipulation of the host's behavior. It's not just draining the crab, it's got its strings plugged directly into the puppet's control panel.
That is deeply unsettling. And what's amazing from an evolutionary standpoint is what the Sacculina's own body tells us.
You mean how simplified it is?
Yeah. It's lost most of the features of a free-living barnacle. It's basically just roots and a reproductive sac. Yet, its genome, its DNA, is apparently just as complex as its free-living relatives, like lobsters or other barnacles.
Exactly. It's retained all the complex genetic machinery, all the regulatory systems needed to manipulate another complex organism, while ditching the need to build and maintain its own complicated, energy-hungry body.
It's evolution by subtraction.
A really powerful example of it, it totally challenges our intuitive idea that evolution always means adding complexity, getting better in some linear way. Sometimes the winning strategy is radical simplification of your own form, coupled with a hijacking of someone else's. Evolution basically asking, why bother growing a whole body when I can just steal yours and make you do all the work?
It's horrifyingly efficient.
That's a good way to put it.
Okay, so this ability of Sacculina to just completely hijack a crab, as disturbing as it is, you mentioned it's not unique, this kind of parasitic mind control happens elsewhere.
Oh, absolutely. The natural world is teeming with examples that are just as bizarre, sometimes even more targeted. If you thought the barnacle was wild, brace yourself. We're going to broaden out now and look at a few other biological puppeteers. Keep that idea of the erased self in mind.
All right, I'm ready. Hit me.
Okay, let's start with one that sounds like pure nightmare fuel. The lancet liverflick. Dicrocilium dendriticum. It's a tiny flatworm with a seriously complex life cycle.
Where does it start?
It starts in snails. The snail eats the eggs, the parasite develops, and then the snail excretes the larvae in these little slime balls.
Gross. Okay.
Then ants come along, and for some reason, they eat these slime balls. So the ant becomes the second intermediate host. Now, here's where it gets diabolical. Usually, hundreds of fluke larvae infect the ant, but one, just one, does something different.
What does it do?
This single larva migrates up into the ant's head, and it attaches itself directly to the nerve center that controls the ant's mandibles, its jaws. It basically takes control of the jaw muscles.
Oh, no. So what happens to the ant?
Instead of going back to the nest at night like a normal ant, the parasite forces the infected ant to climb up a blade of grass at dusk. And when it reaches the top, the fluke makes it clamp its mandibles down hard onto the grass tip.
What does it stay there?
Stuck all night.
Stuck. Immobilized. Like a tiny zombie flagpole topper, waiting.
Waiting for what? Waiting to be eaten. That position, high up on the grass, makes the ant incredibly easy prey for grazing animals like sheep or cattle. And those grazing mammals. They're the fluke's definitive host. The fluke needs to get inside one to reproduce sexually. The ant is just a taxi service to the final destination.
That is horrifyingly precise. What if it doesn't get eaten?
That's maybe the weirdest part. If the ant survives the night, the warmth of the morning sun seems to trigger the fluke to release the cramp. The ant unclamps its jaws, climbs down, and goes about its day like nothing happened.
Seriously, it just resets.
It resets until the next evening when the compulsion takes over again and it climbs back up the grass, night after night.
Right.
A tiny, horrifying biological time bomb forcing the ant up its grassy execution post until it finally gets eaten.
Wow. Okay. What else you got?
Well, there's Toxoplasma gondii. This one's a protozoan, a single-celled parasite. Its intermediate hosts are usually rodents like rats and mice. And its definitive host, where it reproduces sexually, is cats.
Ah, Toxoplasma. I've heard of this one. Doesn't it make rats less scared of cats?
Exactly. It famously makes infected rats lose their natural innate fear of cats. In fact, some studies show they can even become attracted to the smell of cat urine, which normally sends them running for the hills.
So it makes them easier for cats to catch?
Precisely. It increases the chances of predation, ensuring the parasite gets into the cat's gut to complete its life cycle. What's really fascinating is how it might do this. Toxoplasma shows a distinct affinity for infecting the amygdala in the rodent brain.
The fear center.
The fear processing center, yes. It's literally targeting the part of the brain responsible for the host's survival instincts regarding its main predator.
How does it change the fear response?
Well, the exact mechanisms are still being unraveled, but studies have found it can increase dopamine metabolism in certain brain areas. Dopamine being a key neurotransmitter involved in reward, motivation, and movement. So maybe it makes the cat smell rewarding, or just makes the rat bolder, less inhibited.
And isn't there a connection to antipsychotic drugs?
Yes. Intriguingly, some research has shown that antipsychotic medications, which often target dopamine pathways, can actually reduce these toxoplasma-induced behavioral changes in rodents. It really highlights the deep links between parasites, neurochemistry, and behavior, and even raises questions about subtle influences on our own brains, as humans can also be infected.
Okay, that's unsettling on a whole different level. Keep going. More weirdness.
All right, how about the horsehair worm, Spinochordodes tillani?
Yeah.
This thing is truly bizarre. It starts life in water. Larvae get eaten by aquatic insects, which then get eaten by terrestrial insects like locusts or crickets.
So it ends up inside a locust.
Yes. And inside the locust, this worm grows to an incredible size, often much longer than the host itself, coiled up inside. When the worm is mature and ready to reproduce, it needs to get back to water.
But the locust lives on land.
Exactly. So the worm manipulates the locust's brain. It drives the poor insect towards water, a pond, a stream, even a swimming pool, and compels it to jump in.
It makes the locust commit suicide.
It looks exactly like suicide. The locust plunges into the water and drowns. This allows the adult worm to wriggle free from the dying host and swim away to find a mate in its required aquatic environment. Talk about commandeering your ride and then forcing it off a cliff.
That's grimly effective. Okay, one more. Give me peak visual horror.
Pink visual horror. Okay, I give you Leucochloridium paradoxum, the green-banded broodsac. It's another type of flatworm, a fluke. Its intermediate host is a snail.
Snails again. They get a raw deal.
They really do in the parasite world. So the larval stages of this fluke migrate into the snail's tentacles, the eye stalks.
Okay.
Once inside the eye stalks, they dramatically transform them. The tentacles swell up, become brightly banded with green and brown, and then they start to pulsate rhythmically, visibly.
Pulsating eye stalks. Why?
Because those swollen, pulsating, brightly colored eye stalks look uncannily like juicy, wriggling caterpillars or grubs, the perfect snack for a bird.
And the bird is the final host.
The bird is the final host. The parasite essentially turns the snail's eye stalks into irresistible, flashing lures. Birds see what looks like an easy meal, peck off the tentacle, or eat the whole snail, and ingest the parasite, completing its life cycle. The snail is turned into a living fishing lure for its own predator.
Okay. Wow. Pulsating eye stalks. That's definitely memorable. So these aren't just random side effects of being sick, are they? This is targeted manipulation.
Absolutely. These are generally considered adaptive manipulations that clearly benefit the parasite's transmission and reproduction, not just pathological symptoms. The parasite is actively reprogramming the host.
And you mentioned they target key systems, neural endocrine.
Right. They can target 4 main physiological systems that shape behavior. The neural system itself, the endocrine system, hormones, the neuromodulatory system, things like dopamine and serotonin, and the immunomodulatory system, influencing the immune response. And crucially, these systems are all deeply interconnected, which makes pinpointing the exact mechanism really tricky, but also explains how parasites can cause such widespread effects.
So how do they do it? Are there different levels of sophistication?
Definitely. Sometimes the manipulation is relatively simple, almost like a brute force method. Just draining the host's energy can work.
Like the malaria example.
Exactly. Mice with malaria get feverish, lethargic.
Yeah.
They just don't have the energy to avoid mosquitoes effectively. The parasite wins by simply weakening the host. Or think about that tapeworm, Echinococcus granulosus. It forms huge cysts, often in the lungs of moose.
That can't be good for breathing.
Not at all. It likely reduces lung capacity, makes the moose weaker, slower, and thus easier prey for wolves, which are part of the tapeworm's life cycle. It's manipulation through debilitation, leading to general vulnerability rather than a specific behavioral change like climbing grass.
Okay, so that's the less subtle approach. What about more direct targeting?
Then you often see parasites targeting the central nervous system, the CNS. It's the command center, so it's prime real estate for manipulation, either through physical damage or more subtle chemical interference.
Like the eye fluke.
Diplostum spathicum, yes. It insists right in the lens of the fish's eye, causes cataracts. The fish can't see well.
So it behaves differently.
Right. It tends to swim nearer the surface, perhaps because it's harder to navigate or find food deeper down with cloudy vision. But swimming near the surface makes it much more visible and vulnerable to predatory birds. It's like, as one scientist put it, a psychiatric equivalent of a lobotomy. Not direct mind control, but altering behavior by messing with a key sensory input.
And the killfish parasite, that one was in the brain, right?
Euoplorchus californiensis, yes. That one insists directly on the brain of the California killifish. And it causes really conspicuous swimming behaviors, jerky movements, swimming near the surface, flashing their underside.
Making them super obvious to birds.
So obvious that infected fruit are estimated to be 30 times more likely to be eaten by birds, the final host. This might work by suppressing the fish's natural stress response, making it less cautious. And significantly, the parasites, the Metasecariae, tend to cluster right on the fish's brainstem, a critical area for controlling movement and basic functions. They're not just randomly scattered, they're positioned for maximum impact.
Okay, so we see these correlations. Parasite is present, behavior changes, transmission happens. But you mentioned the missing link is often proving direct causation.
Exactly. For a long time, much of the research could only show correlation. Parasite A is in host B, and host B behaves strangely, C, which helps parasite A get to host D. But proving that A directly causes C via some specific mechanism, B, that's the hard part.
We need the how.
We need the how. We need to link the parasite's action, event A, directly to the host's physiological change, event B, that results in the behavior, C. Think about tobacco addiction again. Smoking tobacco, event A, causes nicotine release. Nicotine hits specific receptors in the brain, triggers dopamine release leading to addiction, event B. Nicotine is the causative factor. We know the molecule, the target, the pathway. We need the parasite equivalent of nicotine. Precisely. And we do have some good examples. The jewel wasp and the cockroach is one of the best understood. The wasp stings the cockroach directly in the brain, event A. The venom contains specific neurotoxins that target octopamine neurons, the mechanism. This disrupts the cockroach's ability to initiate movement on its own, event B, making it a passive zombie the wasp can lead away. The venom components are the clear causative factors.
So the goal is to find these manipulation factors for other parasites.
Yes. These are the specific molecules or substances released by the parasite that directly mess with the host's normal physiology to change its behavior. Identifying these factors and figuring out where the parasite makes them, is it a special gland, an enzyme? Are they repurposing their own cellular machinery? That's the cutting edge of this research.
How do scientists even look for them?
A promising strategy is a bottom-up approach. You look at the parasite's gene expression, which genes are turned on or off during the specific life stages when manipulation is happening. Then you try to identify the proteins or other molecules produced by those switched-on genes. Those molecules become your prime suspects for being the manipulation factors. It helps distinguish deliberate manipulation from just, the host feeling generally unwell because it's sick.
And does the size difference between the parasite and host matter here?
It likely does, in terms of delivery. A tiny parasite trying to control a huge host might need to produce and store concentrated doses of manipulation factors, maybe inject them strategically. Whereas in systems where the parasite is closer in size to the host, maybe a continuous high rate of production is enough to maintain control. It adds another layer to how these incredible manipulations can be achieved across such diverse pairings.
So beyond direct damage or energy drain, what are the really sophisticated methods? You mentioned neuromodulators and the immune system.
Right. This is where the control gets incredibly nuanced. Parasites can achieve really fine-tuned manipulation by producing neuromodulators themselves, or perhaps more commonly, by tweaking the host's own neuromodulatory system, suppressing or activating them.
These are things like dopamine and serotonin.
Exactly. Monoamine neurotransmitters like dopamine, serotonin, and in invertebrates, octopamine. These aren't just simple on-off switches. They act more like dimmer switches, influencing mood, activity levels, stress responses, social interactions, reproduction, a whole suite of complex behaviors. And because these systems are ancient, evolutionarily conserved across both vertebrates and invertebrates, they provide common targets for parasites to exploit, regardless of the host.
Can you give an example?
Sure. Take those spiny-headed worms. Echincanthoscephalans like Polymorphus paradoxus infecting gametids, those little crustaceans.
The ones that cling near the surface.
That's them. They become attracted to light, positive phototaxis, and cling to surfaces near the water surface. Both make them easy pickings for ducks and other waterfowl, the parasite's final hosts. This whole behavioral shift is strongly linked to changes in the gametid's octopamine OA activity.
Yeah.
And get this, if you experimentally inject octopamine into uninfected gametids, you could actually suppress that clinging response. It's strong evidence for OA's role.
Wow. And serotonin.
Serotonin, or 5-HT, is also heavily involved. In some gametid infections, being attracted to light is linked to elevated serotonin levels in the brain. But in others, like infections with Microphallus papillorobustus, which physically invades the brain, you see lower levels of serotonergic activity. It shows how complex and context-dependent these neurochemical manipulations can be. It's not always the same switch being flipped.
And dopamine seems to pop up a lot too.
Dopamine DA is definitely a key player. We mentioned the shore crabs infected with Prophylocolus antarcticus. They become hyperactive, linked to increased dopamine in their hemolymph, their blood equivalent, and the killifish with Euoplorchus in their brainstem show increased dopamine activity, likely contributing to that risky swimming.
Didn't you say toxoplasma might even make dopamine-related enzymes?
Yeah, remarkably, toxoplasma has genes for tyrosine hydroxylase, the enzyme needed to synthesize dopamine. It suggests it might be actively producing or boosting dopamine directly within the host's neurons. And again, the fact that dopamine-targeting antipsychotics can reduce toxo's behavioral effects in rats is just, well, it really underscores the power of hijacking these core pathways.
It's like finding the master controls for behavior. But what about the immune system? How does that fit in?
That's another incredibly sophisticated layer. Parasites can leverage the host's own immune responses, particularly neuroinflammation inflammation in the brain or nervous system. The brain's immune activity, things like nitric oxide levels, can directly influence neuromodulatory activity. So a parasite might not need to make dopamine itself. It could just trigger a specific immune response that then causes the host's brain to alter its own dopamine levels.
An indirect manipulation.
Exactly. It's using the host's own defense system against itself. Remember, the Euoplorchus sits on the killifish brainstem. Researchers found they express fibroblast growth factors nearby.
What do those do?
Fibroblasts are involved in healing and scar tissue formation. Why the parasite is stimulating these cells right there on the brain isn't entirely clear. Yet, maybe it's to encapsulate itself safely, maybe it's part of the manipulation pathway, but it shows the parasite interacting directly with the host's cellular repair and immune mechanisms right at the site of control.
So it's all interconnected nerves, hormones, immune cells.
Massively interconnected. There's extensive crosstalk between the neuroendocrine system, nerves and hormones, and the immune system. A parasite hitting one point can trigger ripple effects, cascade effects through these linked systems. It allows for incredibly complex, multi-layered behavioral changes with potentially minimal direct effort from the parasite's perspective. This whole integrated neuroimmunoendocrine system becomes the playground for parasitic manipulation.
It's just an astonishing level of biological control. Mind-boggling, really.
It truly is.
So as we kind of wrap up this deep dive, thinking about Succulina, thinking about zombie ants and fearless rats and pulsating snail eyes, it feels like we have to circle back to that idea of evolution. It really does come across as this, well, masterpiece.
Yeah, masterpiece is a good word.
But not because it's aiming for perfection or creating things that are just good or beautiful in our human sense, but because it just relentlessly finds these ingenious, sometimes absolutely brutal, often deeply bizarre, but always fascinating solutions to the core problems, surviving and reproducing.
Absolutely. These solutions that seem impossible or that defy our intuition, a barnacle consuming a crab's identity from the inside out, a fluke programming an ant's nightly climb, a protozoan dialing down fear. These aren't just weird accidents. They are the results, sometimes messy, sometimes horrifying, but the logical results of variation, selection, and unimaginable spans of time working relentlessly together. They're just testament to the sheer unbridled creativity of life under pressure.
It really feels like there are no simple answers here, does it?
Yeah.
Just profound complexity and endless fascination that just invites us to keep digging, keep exploring these deep dives into the natural world.
Yeah, the more you learn, the weirder and more wonderful it gets.
It definitely makes you wonder about all the hidden forces shaping life around us, even within us, in ways we might not even perceive. Thanks for diving in with us today. It's certainly given me a lot to think about.
My pleasure. It's endlessly fascinating stuff.